Okay, thank you very much, Yuri. I'm going to turn down my phone so that you can hear me through the speaker on my Mac if there's any issue. So first of all, I do apologize for some of the technical issues that we've had and also for not being there at a really exciting meeting. And I'm delighted that the ISMB has, catalyzed this particular session, which will continue, I think, in forthcoming meetings focused on advances in systems immunology. So I just wanted to open with giving you a sense of my own journey. I'm really a molecular biologist by training. And then over several decades, I would say, particularly the last two, have been drawn to systems biology and also, of course, the introduction of various means of using artificial intelligence to understand the complexity of the regulatory and molecular networks that we as experimental biologists are interested in. And over the years, what we've been interested in are really two big problems that I think still are capturing the attention of systems and computational biologists. One problem is, of course, in analyzing the large set of transcription factors, proteins that are directly interacting with DNA sequences in the genome to control very, very complex patterns of gene expression, both spatial and temporal. And then the secondary and related problem is how these regulatory proteins are interacting with one another in the genome in the context of complex information processing circuits to regulate cell fate and cell functions. And so we began by, discovering many of these transcription factors individually using biochemical and molecular genetic strategies, and then sort of like others, linking them to target genes and elaborating those networks. And it is that kind of painstaking work, experimental work, that led us over time to assemble many of these regulatory proteins into what I would call simple gene regulatory networks. And on this slide, I'm just illustrating two of the networks that we assembled some time ago. The one on the left is regulating cell fate decisions within the hematopoietic system, controlling the generation of macrophages or B cells of the immune system. And one on the right is regulating a bifurcation that B cells undertake when they're activated to give rise to short-lived antibody-secreting plasmablasts or germinal center B cells that undergo this fantastical process of somatic hypermutation and affinity maturation, generating clones that secrete higher affinity antibodies to the antigen that instigated the response. And as you can see from both of these kinds of analyses, these are simple networks because they typically comprise of 4 to 5 key transcription factors for which we and others had molecular and genetic evidence as to their involvement. They don't really specify the large set of target genes that are controlled by these regulatory proteins that awaited, of course, the advances in a variety of experimental genomic methods. And now, of course, we have the ability to connect these regulatory proteins to hundreds, if not thousands, of regulatory elements and target genes that they're controlling. And this is where I think the advances that are now being made vis-a-vis artificial intelligence and the ability to recognize very, very complex patterns and circuits positions this arena in a very exciting period. So I want to spend the rest of my talk really talking about elaborating on one of the major problems that still, I think, is of fundamental interest, and that is what is the nature of the lexicon of the transcriptional regulatory code? And by that, I mean the set of DNA sequences that are recognized by transcription factors, not just acting alone, but in a combinatorial capacity to in turn dictate very complex patterns of gene expression. And I just want to remind you that there are two fundamental processes or mechanisms that we consider when we're thinking about the control of gene activity in complex metazoan genomes. One is that these regulatory sequences that control gene activity are, by their very nature, involve combinatorial control. So you have complex combinations of binding sites for these transcription factors that are deciphered to control gene activity. And the second is that you have extraordinary action at a distance. Unlike in prokaryotic genomes, where all regulatory sequences, or the vast majority of them, are positioned very close to the transcriptional start site within 100 or so base pairs, with few exceptions. Of course, in metazoan genomes, these regulatory sequences can be positioned at very, very large distances over a megabase from the transcriptional start site. So both action at a distance and combinatorial control I see as two fundamental mechanisms where metazoan genomes have evolved away from the constraints for controlling gene activity that are inherent in simpler genomes. And these freeing up these constraints, I think, actually enabled metazoan evolution, because it enabled these genomes to have much more diverse patterns of gene activity, which were needed for cellular specialization and functional capacities. So now to come back to the problem of the nature of this transcriptional regulatory code, we've known for some time that the building blocks are these short 6 to 10 base pair DNA motifs. They're redundant, and they have considerable redundancy. They represent binding sites for individual transcription factors. And you can draw an analogy with amino acyl tRNAs interacting with codons in the context of the genetic code. But it turns out that there's considerable combinatorial complexity to the code. As I mentioned earlier, that each of these regulatory sequences, when we've analyzed them, turn out to have diverse combinations of motifs. And so the higher order organization of these motifs, I think, remains of intense interest. And so from the simple motifs, you can generate a lexicon, and from the lexicon, you can generate a syntax. These words are used quite frequently to talk about the nature of the code. And you could even draw an analogy with language, with words, phrases, and sentences. And I think this is where we need deep learning to help us figure out the nature of this regulatory language. Now, if you think about the approaches that are being used or have been used to decipher the code, molecular biologists like me started, with a mechanistic framework and tried to build up the nature of this code using a bottom-up approach. Of course, statistical genomics has been widely used subsequently based on the availability of large-scale genomic profiling data sets. And then finally now, computational genomics based on deep learning is being used to address this problem. So I want to share with you an advance that we've made in identifying what we think is a predominant layer of the code, which consists of composite DNA elements. And by composite DNA regulatory elements, I mean stereospecific juxtapositioning of 2 or more transcription factor binding sites. And in the stereospecific juxtapositioning, the spacing of the sites, as well as their relative orientation, if the motifs are not palindromic, matters. And of course, you can imagine that if you have these stereospecific juxtapositionings that are widely used in the context of the code, they allow you to enable combinatorial control of activity. The simplest thing to consider is that they allow integration of signaling inputs. If you have two different transcription factors that are each independently regulated by two different signaling inputs, A and B, and they cooperatively bind to one of these composite elements, of course, that's a very simple way of integrating two distinct signaling inputs to control genome activity. So these are exemplars of composite transcriptional elements that we and others have described. And if you look in the literature now, you would probably find 25 to 30 or so well-described examples of this sort. In each case, what you can see is that the composite elements are formed by juxtapositioning the binding sites for individual transcription factors, the spacing of the binding sites, as well as their relative orientation is conserved. And one can demonstrate, as we did many years ago, cooperative DNA binding, using biochemical assays with the individual regulatory proteins. And you can demonstrate in those biochemical assays that changing the spacing of the sites or their relative orientation abolishes cooperative interaction, biochemical cooperative interaction, as well as cooperative function when you analyze transcriptional activity. So given these 25 to 30 well-described examples of composite elements, one can take the following two views. One is that these composite elements, in spite of their significant interest from a molecular and biological standpoint, are outliers within the larger regulatory code. In other words, the regulatory code is dominated by combinations of simple motifs which do not necessarily have these kinds of stereospecific juxtapositioning rules. Alternatively, one can take the view that actually the second layer of the code involves a very large set of composite elements. And the reason we have not come to appreciate that is simply because we've not elaborated systematic approaches for analysis of such elements. And I'm going to argue the latter, that in fact composite elements are a dominant second order feature of the regulatory code. They're equivalent to the phrases. And in the next 15 minutes or so, I'm going to elaborate a framework that we've used for systematic discovery, testing, and learning of this lexicon. And this involves three different aspects. The first aspect is using statistical genomics to analyze a very large set of regulatory DNA sequences in the immune regulome to discover what I would call candidate composite elements. The second part of this approach is an experimental arm where we have designed a custom MPRA library that brings in all of the predicted candidate composite element sequences from the first step and tests them for their activity as activating sequences. And then the third is using a deep learning model to learn the relationship between these DNA sequences that carry the composite elements and their transcriptional activities, such that we can, at base pair resolution, predict the activity of any DNA sequence in a context-specific manner on the basis of composite elements that it might carry. So let me just very quickly step through the three parts of this overall strategy. The first uses a statistical genomics pipeline that we've developed called CE-seq. And essentially what it does is it systematically takes a set of test sequences that you provided, regulatory DNA sequences, and searches them for composite elements, that is, stereospecific configurations of any pair of motifs for, let's say, transcription factors A and B. And then it generates outputs where, for example, in this case, we have searched for composite elements comprised of IRF and ETS motifs or IRF and AP1 motifs, allowing for 16 different spacing configurations and all 4 possible relative orientations. And you can see in the middle in this heat map particular configurations that are statistically enriched in our test set versus a background set of sequences that we used. And the pipeline actually generates position weight matrices for the most statistically enriched configurations. Now, in this toy example, the statistically enriched configurations that one sees on the right are ones that we have previously experimentally described. So this only tells you that the method is working to rediscover regular composite elements that have been previously described. We then have used CEC to analyze a very large regulatory DNA space. The ImGen consortium has generated a compendium of regulatory DNA sequences that are operative in essentially all immune cell types, including actually hematopoietic cell types. We've taken all of those regulatory DNA sequences and systematically searched them for candidate composite elements. And when we do that, we end up with predictions of roughly 5,000 candidate composite elements. And I show you 2 of these here on the bottom because they're really quite, actually one of them on the bottom, because it's really an interesting stereospecific juxtaposition of the binding site for STAT5, a cytokine-regulated transcription factor, along with that of RBPJ, which of course is the subunit that transduces notch signaling. So this would imply perhaps that this composite element functions to integrate these 2 different signaling inputs into the genome. So the next step, given this very large set of candidate composite elements that come from our statistical screen, we decided to build a massively parallel reporter assay library that carries all of these composite elements. And the design of that library is shown on this slide, where for each of these composite elements, we also have in the library mutant versions of them where we mutated site 1, site 2, or site 1 and 2, changing base pairs that have the highest information content to complementary. The rule that we followed is the G is mutated to a T, the A to a C. This library additionally contains all simple motifs that we know about and mutant versions. And so the power of this library is the following, that if you introduce it into a cell of interest, essentially what you're doing is simultaneously measuring the activities of all transcription factors that are operating on simple motifs. You can get quantitative measures of those, as well as you're getting the activities of pairs of transcription factors that are acting on these candidate composite elements. So in a sense, it allows you to quantitatively analyze the activities of a very large set of transcription factors, either acting singly or in combinations, and of course, validate which of these CEs are actually operating in particular cells. Now we've introduced this library that contains over 50,000 individual sequences whose activity we can measure, transcriptional activity we can measure, into 3 different cell types. Those are GM12878 cells, human B cell line, K562 cells, an erythromyeloid cell line, and Jurkat T cells as a T cell line. So this is an example of a composite element whose activity is revealed in GM12878 cells. You can see the activity of the composite element alone. Mutating motif 1 diminishes the activity, motif 2 diminishes the activity, and of course both we failed to detect any activity. So you can basically, in one very large assay, and the assay of course generates a huge data set because we're getting quantitative measures of each of these sequences, both the wild type and the mutant sequences, and we can then systematically determine which ones are active and is their activity dependent on the predicted motifs. And so these are examples of newly discovered composite elements in GM12878 and K562 cells. You can get another validation of these composite elements by asking, are these predicted composite elements, which are shown now to be active, are they in fact enriched in binding regions of the cognate transcription factors? So what we've done here is to take ChIP-seq data sets for PU1 and the AP1 family member Jun. We've overlapped those ChIP-seq regions. And so you can see these are the overlapping regions. These are uniquely bound by the two regulators. And if we use CE-seq now to ask if this particular composite element is selectively enriched in the co-bound regions of these two transcription factors, it in fact is. And you can see that these two highlighted boxes in the heat map represent basically that same composite element. And the only reason you have two is that the AP1 motif is a palindromic motif, and so the two configurations represent the two slightly different versions of that sequence when it's flipped. So this kind of analysis argues that not only are the composite element active and their activity is dependent on coordinate action of these two stereospecific motifs, but in fact there is evidence that they're co-bound by the relevant regulatory proteins in vivo on a large set of genomic regions. So by way of example, I just want to show you that if you look at the AP1 motif and its CE lexicon, in other words, other motifs that it's stereospecifically juxtaposed with, this is an example of the diverse set of composite elements that are constituted by the AP1 motif. And I need not emphasize the fact that the AP1 family of transcription factors functions in a diverse set of cell types in response to a variety of signaling systems. Therefore, it's not surprising that it has an elaborate CE lexicon. The same is the case for the KLF family, and one can systematically see how particular TF motifs have been combinatorially expanded through their inclusion in these composite elements. The last part of the strategy involves now learning from these very large, massively parallel reporter assays and using a neural network model to train on these individual DNA sequences and their transcriptional activities, of course, that are measured. So what we want to do in this case is to basically extract the features of the motifs that are contributing to activity, as well as to learn the weights or the interaction rules amongst the motifs. So the model has to be aware of spacing since stereospecific juxtapositioning is a key feature of these CEs. And so the model is termed GRACE, and it in fact does actually quite a good job in terms of once it's trained on a set of DNA sequences and the activities that we measure, it can now make single base pair resolution predictions. And by way of example, here is one of the sequences that was used in our MPRA library. And this is the position weight matrix that it was based on from the CE output. So using GRACE, we perform in silico saturation mutagenesis, systematically varying each of the base pairs. And of course, GRACE predicts the activity of the mutant sequences. And then from that analysis, we can generate a contribution score matrix that is analogous to this position weight matrix. So this is, I should emphasize, this is predicted by GRACE, whereas this is, of course, the statistically enriched sequences in the gene. And I think you could see that GRACE does actually a very good job in terms of being able to learn at base pair resolution which of these positions are particularly critical for accounting for the activity that we measure. Now, I want to end with just pointing out two other things that GRACE is able to do. Once GRACE is trained on context-specific data sets, and by context-specific, I mean we have three different GRACE models, one for B cells, one for T cells, and one for erythromyeloid hematopoietic cells, based on the three different MPRA data sets. Now, for any given DNA sequence, we can actually make a prediction vis-a-vis what its transcriptional activity is likely to be in these three different cellular contexts. And so in this case, what we've done is to use the GRACE model to predict the transcriptional activity of open chromatin regions in these human B cells and just rank ordered them. And then we've asked, okay, how well do they compare with their accessibility as measured by ATAC-seq? And you can see that there's a reasonable relationship between the ordering of their transcriptional activity and the ordering of their accessibility. And there's a reasonable relationship between this activating histone modification, which is important for transcriptional activity as well. So in this kind of coarse-grained way, GRACE predictions line up with chromatin accessibility and activating histone modifications. But GRACE actually is able to make a fine-grained predictions. I'll remind you that transcriptional enhancers in the genome are known to promote bidirectional transcription. So given that, we asked if we take DNA sequences that reside within open chromatin regions and make GRACE predictions of where GRACE is predicting the active DNA sequences are, how would those predictions line up with actually measured bidirectional transcription at enhancer elements? And so these are predictions that we've, and their testing that we've carried out in the human B cell line. And so these blocks represent DNA sequences flanking open chromatin regions that are enhancers because they show bidirectional transcription as one of their features. The red areas indicate GRACE predictions of transcriptionally active sequences in these groups of enhancers that can be aligned on the basis of the regions that are predicted by GRACE to be transcriptionally active. And these, and the bidirectional transcription that's emanating from these enhancers is drawn from encode data, which is actually GROCAP signals. And so what you can see as you go down these groups of enhancers in the genome is that GRACE predictions typically lie within regions of bidirectional transcriptionome that are measured at these enhancers. And so we think this is a striking example where even though GRACE was trained on MPRA data, transient transfection assay data, it is able to make predictions about the activity of regulatory sequences in the context of chromatin in the genome in a context-specific manner. And I want to end with one final comparison. I think you're well aware that, you know, at this point in time, we have deep learning models that can be trained on chromatin structure, specifically chromatin accessibility and ATAC-seq data, and they can make base pair resolution predictions vis-a-vis the sequences that are accounting for chromatin accessibility. And I think Anshul Kundaje probably has presented ChromeBPNet at the meeting, and so it is an example of one of these deep learning models that can be used to make predictions around chromatin accessibility. So I would argue that GRACE actually complements models like ChromeBPNet because our model is trained on DNA sequences and their transcriptional activity rather than DNA sequences and their chromatin accessibility. And so an outstanding question is, how well do these two models do in terms of deciphering the nature of the underlying transcriptional regulatory code? Not just simple TF motifs, but of course composite elements as well. And so here I show you an example of this, where these are PWMs of the simple motifs that comprise a composite element, shown in the middle. And in the second row indicates the predictions by Chrome BPNet. Now, Chrome BPNet is able actually to capture the composite element, although its predictions in terms of the simple motifs are not quite as good. Grace is able to predict or capture both the simple motifs in this data set and, of course, predict the composite element as well. Now, one way to interpret this data would be to suggest that perhaps in this case, the individual transcription factors contribute minimally to chromatin accessibility, but their combinatorial binding to the composite element enables opening up of regions that carry the composite element, as well as, of course, activating transcription through them. This is a systematic analysis of all of the Grace and Chrome BPNet predictions in the hematopoietic cell line K562 and in GM12878. So in one case on the y-axis, we have the Grace predicted activity. Along the x-axis, the Chrome BPNet predicted activity. By and large, there's actually a very good correspondence. And that argues that both deep learning models are actually learning the same underlying lexicon. And I would argue, therefore, providing compelling evidence for this lexicon, which I remind you is not just simple TF motifs that we've known about, but a lot of these composite elements that we have recently discovered. There are examples, however, of motifs that are preferentially learned by one model. For example, the ones along the x-axis here, or motifs that are primarily learned by the other model. And I think that is an illustration of transcription factors that are primarily, from a molecular standpoint, functioning to either regulate chromatin structure or transcriptional activity. I won't have time to expand on this part, but of course, Grace can be used to make predictions around regulatory variants, specifically GWAS variants associated with various kinds of diseases. And the model can be used to determine, whether a composite element could be the relevant motif that is accounting for altered gene expression that might be associated with a particular disease. So I just want to thank a lot of people that have actually enabled this analysis. In particular, this has involved a large collaboration with colleagues at MD Anderson, Nidhi Sawhney, Dana-Farber, Cliff Meyer, and at the Jackson Lab, Ryan Toohey, who helped to construct the custom motif library, and John Ray at the Benaroya. So thank you very much, and I'll be happy to take some questions.
Thank you, Harinder. I don't know if he still cannot hear us or he can now. Harinder, can you hear us? Okay, I think I will need to then use a backup system of calling him on the phone. Hello, Harinder. He cannot hear us except on the phone. Is it correct?
Yes, that's correct.
Okay, so we will use these remaining 2 minutes for questions, and we will use this through my phone. So there is a question from the audience, Ferhat.
Okay. I'm close now.
Yes.
Speak on the one.
But also on the mic here, so that
this is Farhad.
Ferhat, also for the audience.
Yeah, for the audience. Yeah. Okay. So my question is about more recent work from Chris Benner and Sven Heinz, kind of like making things a little bit more complicated with respect to like the transcription factor motifs and how they are positioned with respect to transcription start sites, having like very significant effect on their activity or like direction of activity essentially. which I assume applies to composite motifs too, and is not captured by the MPRA. Just want to know what you think about that.
Yeah, no, I know the paper that you're referring to, and I agree that actually those kinds of I think we're looking at sort of two different aspects here. Although where there is convergence is in that last set of analyses that I showed. What I argued was that the sequences that Grace is predicting are accounting for transcription. Actually, what is interesting to us is that it is also able to predict where transcription is initiated. And that's not just the bidirectional transcription that is seen at enhancers, but we have a similar analysis of transcription at all active promoters in a given cellular context. And so, you know, I think it's a fair point that you raised, though, that we need to try to integrate our analysis with work that Chris and Sven, recently published. Although I don't think that there's any kind of inconsistency. I think we're emphasizing two different aspects of the problem. In our case, we're trying to emphasize that stereospecific positioning of these motifs is a major feature of the underlying regulatory sequences. It just has not been appreciated to the extent that I think it needs to be.
All right, thank you. I think I will ask very brief question, Harinder. So, not as a concept, but specific sites. I'm very interested in all what you presented, but just one aspect. So, how deeply conserved this is, because it's encoded in the sequence. So I can assume that these juxtapositions of specific motifs can be broadly conserved between many different species. Is there a way to say something about it?
Yeah, I think this really needs to be pushed quite a bit. Or I mean, the evolutionary considerations I think are extremely interesting. All we can show is because, and I skipped over this, that we've gone back and forth between the murine and human genomes, and for all of the several thousand composite elements that we have, we see conservation of the spacing and the relative orientation in these two genomes. But of course, one should undertake a much wider evolutionary sweep. And this is of great interest to me because the first set of composite elements that we described between IRF, interferon regulatory factors, and the S family factors. Well, it turns, I think, the proteins that cooperatively bind appear to have arisen relatively recently in the context of metazoan evolution. And I think what stands to reason, therefore, is that the composite elements that they recognize are likely to be a more recent emergence. But we haven't systematically explored this, and I thank you for raising that issue.
Okay, thank you very much. I think we are short on time. I know there is at least one nice question online. If you can actually answer it also online, it will be great. And thank you very much again for this wonderful talk. We need to move on with the program. Thank you.
Okay, thank you. Bye. Bye.
Bye-bye.